Delighted to introduce our first speaker, Dr. Rick Karban. Dr. Rick Karban is a professor in entomology from the University of California, Davis. He got his bachelor's in environmental studies from Haverford College and his PhD in ecology from the University of Pennsylvania. Dr. Karban is nothing short of a titan in his field. He's published over 200 papers. He's written several books, including books on plant communication and induced herbivory. He's a fellow in the Ecological Society of America and the American Association for the Advancement of Science. His current research focuses a lot on volatile organic compounds, plant communication, and primarily uses sagebrush currently as his model organism. He also does quite a bit of research into the abundance and distribution of woody bear caterpillars. But I mean, outside of just his own research, it's clear that Dr. Karban has a passion for helping young ecologists understand how to conduct ecological experiments and be successful in the world of ecological research. He's actually written a book, How to Do Ecology, which I think now is in its second edition, that teaches, designs, and helps young ecologists on how to do research in their field. So without any further ado, Dr. Karban is going to be presenting plant communication today, and I'm delighted to hear what you have to say. So come on up, we're looking forward to it.
Thank you for that introduction, and thank all of you for coming to the symposium. This really is an impressive event and an impressive gathering of ecologists. Thanks. What I hope to do this morning is to convince you that plants are not just organisms that kind of sit there and do nothing, but in fact, they're quite interesting. They behave, and they're quite capable of perceiving things that are going on around them and responding to the information that they receive and communicating with one another. So that is kind of the title of my talk, and that's my take-home message. Okay, I'd like to see if this works. Cool. I'd like to thank and acknowledge the collaborators that I've had who have helped to do the work that I'll describe for you today. Kari Shiojiri, James Bland, Patrick Groff-Tiza, and Satomi Ishizaki. So in my talk, I'm going to address 5 questions. As I said, I'm going to try to convince you that plants can perceive their environment and communicate. And this has been quite controversial. So let me start off by clearly defining what I mean when I say that plants are capable of communication. I'll then consider whether plants communicate about herbivory, about their risk of being attacked, why plants might be perceptive to cues about herbivory, why they might emit cues about herbivory, and that will lead us finally to consider how it all works, who communicates and how they do this. Okay, so what do I mean by communication? The definition that I will use, and I'm fine if you have a different definition that you prefer. I don't care much about definitions in general, as long as we're clear with one another about what we mean when we use these terms. So when I use the term communication, I'm thinking about a process in which an individual, a sender, emits a cue. Whether or not it does that intentionally, I have no idea. And I think that's quite a different kind of scale of problem to address. But a sender emits a cue that's perceived by a receiver and causes that receiver to change in a way that is rapid and conditional and changes its phenotype. That's all I mean by communication. And I'm not making any assumptions about the adaptive value of either of these, either being a sender or being a receiver, although I'm very interested in those questions. Okay, so to some extent, in particular, I'm interested in communication about risk of herbivory. And like I say, this is a little subdiscipline that has been quite contentious in ecology. And I think that there were early reports of this in the 1980s and several influential reviews, and that most ecologists felt like, oh, we've heard of this, we've considered it, we've debunked it, let's move on. This is kind of junk science, and we're done with it. And it's not clear to me exactly why this idea, either that plants can perceive their environments or be responsive to cues from other individuals, has been so controversial. Why all the fuss? So we know anybody who's grown a houseplant and puts it in the window knows that that houseplant is very perceptive to levels of light, for instance, and tobacco plants that are out in the field. If they have sufficient light, they'll kind of spread out and grow into bushy plants. If they have insufficient light, instead their phenotype is they become much more elongated, and they are quite responsive to the quality and the quantity of the light that they're receiving. So we've known forever that plants are able to perceive environmental cues. And the same is also true of what's going on underground. So in very early experiments, this particular set of experiments involving barley plants, these are the roots of barley plants. The plants, or the roots on the left there, were exposed to nutrients throughout the soil medium that they were growing in. The plants on the right were exposed to nutrients just in the narrow band, but between those two dark lines. And sure enough, as we might expect, the plants on the right proliferated roots in the soil region where there were more nutrients. Plants are aware of their environments and respond to their environments. And I'd like to convince you that plants are also, that they're quite perceptive and that they're aware of cues about their risk of herbivory as well. So let's consider the second question. Do plants communicate with one another about herbivory? And in order to do this, I set up this sort of basic experiment with sagebrush, with pairs of sagebrush plants. And this experiment has 3 treatments. Here in the upper left, I've got a pair of sagebrush plants that are growing close to one another. I clip the leaves of one of those plants, the one on the right, and then I measure the damage over the growing season that the assay plant on the left receives. So this is the treatment that I refer to as clipped. The treatment on the upper right there is similar. I call it clipped and bagged. I do the clipping and then immediately enclose the clipped branch in a plastic bag, which minimizes the release of volatiles from the clip branch to the assay branch. On the bottom, I have a control. I don't do anything to those plants. And for all three of these treatments, I measure the amount of damage at the end of the season on the assay branch, the branch that has not been messed with in any way, and just see how much damage does that assay branch receive. And what I find is that the level of damage, and most of my result slides will have the same y-axis, and that's damage to this assay branch. So how much herbivory at the end of the season does that branch receive? And what you can see is that those assay branches that are close to other branches on another plant that have been experimentally clipped experience less damage by herbivores relative to the control, or relative to a situation where the clip branch was immediately enclosed in a plastic bag, minimizing the exchange of volatiles, suggesting that a volatile cue is required. We've done other experiments that I'll describe later on in the talk, where we just move the volatiles from one plant to the other. And that's sufficient to produce this reduction in herbivore damage. So we think that a volatile is required and is sufficient to get the assay branch to change in a number of ways, to change its phenotype in a number of ways that make it less good as a host to its herbivores. We've now done this kind of basic experiment lots of different times. The photo on the left is just a picture of one of my field sites. These sagebrush plants are often neighbors. They're quite abundant in the Great Basin. They're intolerant of fire, and they do really well in places that are overgrazed. Two treatments that we have subjected the American West to quite a bit. We've, pretty much suppressed fires for the last century and overgrazed. And so sure enough, we have an enormous area that is dominated by sagebrush in many places. It's almost a monospecific sand. So we often find individuals of sagebrush growing quite close to one another. We can document this communication between individuals up to about 60 centimeters. So over a relatively short distance, but most individual sagebrush plants have neighbors that are within 60 centimeters. So this is something that probably is quite common in nature. We find that young plants are both the best emitters and receivers of Q. And in the experiments that I described, we did the treatments by using scissors to clip these plants. We've also done similar treatments by caging real herbivores, beetles or grasshoppers, on the plants to do the damage. It's a lot more work than using scissors, but it gives us qualitatively the same kinds of results. Okay, so if this communication is really going on between plants, why might plants be responsive and perceive these cues? Could this be something that has been favored by natural selection? And if so, why? So this cartoon describes an experiment that's similar to the one that I showed you a few minutes ago, except instead of using pairs of plants growing close to each other, we've done these experiments just using individual sagebrush plants, but different branches of individual sagebrush plants. And as in the earlier experiment, this one has 3 treatments. One treatment where we split the leaves of these clip plants on the left here. Another treatment where we do the clipping and then immediately put a plastic bag around the clip plant, around the clip branch, reducing airflow between the clip branch and the assay branch, and then our control that we don't mess with. And as in the previous experiment, what we find is that those branches that have access to the volatiles from the clip branch have reduced levels of herbivory over the growing season. Those branches that are clipped, but in this case, the clip and bag branch, there's the opportunity for communication through the vascular system of the plant. The 2 branches are close to one another, but the clip and bag treatment, where there's the potential for vascular communication, still shows high levels of herbivory, suggesting that for even systemic induction among the branches within an individual, a volatile cue is required. These plants are, I was quite surprised at how poor they were at coordinating among branches. I have since learned that many other plant biologists are not surprised by this result, that plants, especially plants growing in arid areas, have much less of an ability of communicating through their vasculature than, for instance, we do, where all the tissues of our body are connected by the vasculature, our bloodstream. That is much less effective. These vascular connections are much less effective in the case of plant communication. So one reason that plants might emit volatile cues is to coordinate their own systemic responses to threats such as herbivory. We've also attempted to assess the fitness of individuals that have been provided with these cues and respond, or haven't been provided with these cues. And that's a little bit of a difficult task for a system like sagebrush that is a slow-growing, long-lived plant. It rarely reproduces. So in working in this system for 25 years, I've only seen 3 recruitment events at my field site, but 2 of those were in 2010 and 2011. So during those 2 years, we kind of dropped the other things that we had planned and followed the fates of a lot of seedlings. And what you can see is that almost all of the seedlings died during the first summer, but seedling survival was about 10 times higher in both years for those seedlings that had clipped neighbors, the dark circles relative to those seedlings that had unclipped neighbors. So seedling survival is enhanced by being exposed to these damage cues. We've also attempted to keep track of what happens to established bushes that are either exposed to experimentally to experimentally provided cues of herbivore damage or controls that are not provided with these cues. And for both the production of new branches and the production of inflorescences, you can see that those branches that are exposed to these cues of damage produce more new branches and more inflorescences than do the controls. Okay, so it kind of makes sense that plants are responsive to cues of the risk of their getting eaten. Does it make any sense for plants to be emitting this information? Presumably, they're competing with their neighbors. So why be providing reliable information to your neighbors about what they can expect? And so I think that trying to understand why plants might emit cues is a little bit of a tougher question. And one possibility is that they leak out, that these cues are not adaptive. And I'm sort of a raving adaptationist, so I don't like this idea, but it is certainly as plausible as any of the other things that I'll tell you about in a moment. They might not be adaptive. The selection might be for plants to be perceptive to what their neighbors are doing, not to be providing that information. And providing that information might just be a consequence of getting eaten. Another possibility is that these volatiles that are released when the foliage is damaged by herbivores repel the herbivores. And there's some evidence that this is the case. So this is certainly a viable explanation. Another possibility is that that the volatiles attract the predators and parasitoids of the herbivores, and that the plant is essentially getting help from a third trophic level, and that might explain why plants are emitting these volatiles, and the fact that their neighbors happen to eavesdrop on those emissions might be somewhat secondary. And there's some evidence that this is the case. So this shows the number of plants. I had a bunch of plants that I checked up on a couple times a day over a several-week period, sort of had a trap line of these sagebrush plants, some of which I damaged, some of which I didn't. And then I recorded how many either predatory bugs on the left of the screen there, or coccinellid ladybugs, lady beetles on the lower right I found. And as you can see, there were more plants that had more of these predators of the herbivores on plants that had been experimentally clipped. I should probably tell you that the important herbivores of sagebrush are quite varied. And for a long time, I was not a big fan of this hypothesis, largely because it seemed to me that the things like grasshoppers that are quite abundant as sagebrush herbivores in this system would not be very strongly affected by these predators. I have more recently seen outbreaks of leaf beetles, and these predators can be quite effective against these leaf beetles. So I've become more of a believer in the plausibility of this idea that the emission of these volatiles might be favored because they attract the predators and parasites of the herbivores. So I think that there's some reasonable evidence for this idea. these volatiles might inhibit the germination of competitors. And so we've done a series of experiments to evaluate this hypothesis. In this field experiment, so almost all of the germination of sagebrush and of its competitors occurs immediately following snowmelt over a pretty narrow window of time. And so in these experiments, we clipped sagebrush during this period of snowmelt and then recorded how many seedlings of either sagebrush or of other plants we found near these clipped or unclipped areas. And what you can see is that the number of volunteer dicots was reduced close to clipped sagebrush branches. We've done more experiments of this sort in the lath house and with potted plants and potted sagebrush plants, and find that clipping did not reduce the germination of sagebrush seeds, but it did strongly reduce the germination of both volunteer dicots and volunteer grasses. The wild dry were seeds that we planted in the pots, and germination of wild dry was also reduced by these volatiles that were emitted by damaged sagebrush. So I think there's some evidence, and in fact, pretty strong evidence that these volatiles inhibit the germination of potential competitors of the sagebrush. Emitting volatiles may allow plants to mount a systemic induced resistance response, to coordinate their own defenses, if you will. And I've already shown you this slide. This is the experiment that we did that asked whether or not plants are able to coordinate their defenses through the vascular system, and the answer seemed to be not very well. A volatile cue was required. So plants might be emitting these volatiles not to communicate with their neighbors, but to coordinate their own defenses against herbivores. And so I think that this is a reasonable hypothesis for why we see communication between different individuals. So thinking about, okay, maybe what's going on in this system is that the evolutionary driver of this communication might really be about plants communicating with themselves, communication from one branch to the next. And that's what has, that's what's driven selection for this system, and the communication between different individuals might just be sort of something that happens to have come about as a byproduct. And that got us thinking about the possibility of individuals maybe communicating with their kin, and perhaps kin selection is involved, and has sort of led to questions about, well, who's really who's communicating here? Who are the players, and how does it work? And that's where we've gone next. So let me describe some of the experiments that we've done. The first question, it is really a question of whether or not plants are able to communicate more effectively with their own tissues than they are with tissues that are genetically different. And in order to test this idea, what we did was we were able to clone branches of sagebrush to make cuttings of sagebrush, propagate them indoors in the greenhouse, and then move them back out into the field. And so in these experiments, we've got an individual, the assay individual, that is naturally rooted in the field, and associated with that naturally rooted assay individual, we have a potted plant that is either genetically identical to the assay plant, which we're calling self, or it's genetically different from the assay plant, which we're calling non-self. And in this experiment, we actually had 4 treatments. One treatment involved clipping the assay plant. So we expected, okay, we know that induced resistance is a possibility. If we clip the assay plant, we expected that it would have lower additional herbivory throughout the season. And then we have a control where we put in another neighboring plant, but we didn't mess with it. We didn't clip it. That served as a control for all the experimental digging and messing around that we did. Okay, and here's what we found for those 4 treatments. The unclipped control had the most damage. That's not surprising. The assay plant that was clipped had very low levels of damage. That's also not surprising. But the really interesting comparison are the 2, number 3 and number 4, on the right-hand side of these graphs. When the neighbor was clipped and the neighbor and the assay plant were genetically identical, the self-treatment, we got quite a bit of reduction in how much damage the assay plants received. When the neighbor was non-self, it was still some reduction compared to the control, but not nearly as much. So plants are most effective at communicating with each other when they are genetically identical. All right, that was kind of a cool result. And it made us wonder if plants that are more closely related can communicate more effectively than strangers. So Satomi Ishizaki developed a system of microsatellites that allowed her to calculate the relatedness of all of the individual plants that were growing in this meadow that we do a lot of our work in. So we know which plants are closely related, which plants are distantly related. And we've done a series of experiments to ask the question of whether or not communication is more effective among closely related plants than distantly related plants. In this series of experiments, we did this cloning business that is a lot of work, and it's difficult to do with sagebrush. And then brought out potted plants back to the field that were either closely related or distantly related to our assay plants. And in these experiments, we found that branches were more likely to be damaged if they received cues from distantly related, from strangers, compared to from kin. This was a lot of work and was pretty difficult to do. And so we started wondering if we could not necessarily move the plants, which really limited our sample size and was a lot of work, but could we just move the volatiles? So in this first set of experiments, we did the clipping in a plastic bag, and then I sucked up the volatiles, walked over to a neighboring plant and blew the volatiles into a plastic bag. And for reasons that are truly not clear to me, people hate this idea. And they're very bothered by the fact that I was the vector of these volatiles. I like this idea a lot because it means that anybody can kind of do these experiments without any kind of fancy equipment. And somebody wrote to me and said, well, actually, that is a terrible idea since many of these volatiles are quite carcinogenic. And I had recommended these to as science fair projects and this kind of thing. It seemed like, okay, I guess that my scientific colleagues don't like this idea, and it's probably a big liability. We have more recently gotten these giant 1 or 2 liter syringes to move around the volatiles. The experiments that I'll describe sort of moving forward have involved just moving the air in these super syringes. When we've done that, moving the air from close relatives or distant relatives, we find much the same thing, that when the Q comes from a more closely related individual, levels of damage are reduced compared to more distantly related individual. So maybe some kind of kin selection could be involved in this process. But in any case, it seems like the fact that the observation that much of the communication is between self-individuals, or at best between kin, between closely related individuals, has several important consequences. It means that communication allows a plant to coordinate its own systemic defenses. It makes it less likely that when these cues are emitted, that the sender will be providing as big an advantage to its unrelated neighbors. It makes it more likely that plants respond to their own kin. And it raises on other kinds of questions. For instance, do closely related individuals share susceptibilities? So for instance, I'm much more interested in the medical history of my dad than I am in the medical history of, American males, because I'm much more likely to share the same sort of susceptibilities and problems that my dad shared, because we share half of our genes than I am with the general population. So do closely related plants share susceptibilities in a similar way? And maybe more fundamentally, what are the cues? What's the language of this plant communication? I'm far from a chemist. And so I've collaborated with both Kari Shiajiri and James Bland are really chemical ecologists. And they have collected the volatiles from clipped sagebrush, run them through a GCMS, and look to see what are the chemicals that are produced when sagebrush leaves get damaged. And depending upon how fancy the toys are and how careful you are in collecting these volatile chemicals, James and Cowrie can detect increases in something on the order of 100 different volatile chemicals. So the potential language is really quite great. And we know from other communication work involving volatiles that often the combinations of chemicals, the ratios of these chemicals can be quite important. So I think that this is a really complex, but the language here is extraordinarily complex and a little bit discouraging. Nonetheless, I think there are patterns that we can see and that Carrie and James have found, and I'll describe a few of those patterns to you. So this next slide, each of these pie diagrams represents a plant, the volatiles that are produced by a single individual plant. And the orientation of these pie diagrams does not represent any kind of spatial orientation in the field. It's just where we put the pies to make this graph, to make this chart. But you can see that, but each of these individual plants comes from the same meadow. And some of the plants are dominated by this kind of orange apricot color that is alpha-thujone. Some of the plants are dominated by this kind of maroonish color that represents camphor. The thujone plants have a little bit of camphor. Almost all of the plants have a little bit of camphor, but the camphor plants, by and large, do not have thujone. But I guess the main message to me is that in this meadow, there are plants tend to fall into one of two groups that are termed chemotypes. They're either dominated, their emissions are either dominated by alpha-thujone or by camphor. And there are a few outliers that have the green highlighter around them. But by and large in this population, plants are of one of these two chemotypes. And we've done similar experiments moving the volatiles between plants of either the same chemotype or different chemotypes. And we find that communication is more effective between individuals of the same chemotype. Furthermore, looking at parent-offspring regression shows us that these chemotypes are highly heritable. Chemotypes might be responsible for some of the effect of plants communicating more effectively with their relatives, with themselves, than with strangers. What else do we know about these cues? Well, we know that some of these cues are quite general. So I started messing around with this system in part because I was the reviewer of this paper that was ultimately published in PNAS by Farmer and Ryan in 1990. And Bud Ryan had done all this work with a career's worth of work with potted tomato plants. He knew a lot about the secondary chemistry of potted tomato plants. And somehow, at that time, it wasn't known whether methyl jasmonate or jasmonates at all were important plant hormones. And this particular paper was important in establishing jasmonate as an important plant hormone, a volatile plant hormone. But somehow they knew that sagebrush was a mega producer of jasmonate. And so they did an experiment in which they incubated cut branches of sagebrush plants in with potted tomato plants. And these two were incubated together in these old school vacuum jars in their lab. And what they found was that potted tomato plants that were incubated with sagebrush volatiles had elevated levels of defenses. And this seemed very cool to me, but it also seemed like, like weird plants like tomato and sagebrush that never grow together in vacuum jars in the lab. It's a cool result, but does this have anything to do with nature? Unclear. In fact, sagebrush and wild tobacco that actually do co-occur in nature will also communicate effectively. But the point is that some of these cues are quite general, such that even communication between different species at times can be found. And some of the cues are quite specific, such as the communication that only occurs between plants that are of the same chemotypes. This is odd. This should have a whole bunch of different chemotypes on it. It is, for some reason, not coming up. Let's see if, oh yeah, there we go. I didn't make this slide. And so it has like these high-tech things like animation on them. Okay, cool. Patrick Groff-Tisa has done some work where he has now evaluated the volatile emissions of a bunch of sagebrush plants from different populations. And he's found that he can characterize not just 2 chemotypes, as I've shown you, but actually 8 chemotypes. And those are shown here. And right above Patrick's picture is his field sampling device. He's got a throughput system, or I don't even know what they're called, that allows him to do a much better job of this, is the bottom line, than the techniques that I've been using. And he can do all of this in the field. And then he takes the samples back and analyzes them with a GCMS. So there are a bunch of different chemotypes. And what's, I think, the most interesting is that some populations have many more chemotypes than others. Populations are variable in how many chemotypes they have, how diverse they are. This has gotten us thinking. So this is, on the left there is a map of California. The green part of the map shows the distribution of sagebrush in the eastern part of California going into Nevada. The pie diagrams show the proportion of different chemotypes at these different populations. So as you can see, some of the populations only have 2 chemotypes. Other populations, such as Rock Creek, have 7 chemotypes. So there's a lot of variation in how many chemotypes different populations exhibit. And one of the things that we've been interested in doing is sort of looking in on the plant behavior literature, since people who study, I mean on the animal behavior literature, since people who study animal communication have been doing this for decades and have made a lot of progress, have come to understand a lot about how animals communicate. And I hope that Dan's going to talk about that in a few hours from now. or aspects of that. And he's certainly one of the people who's been involved with this progress in animal communication. But it seems to me that there's a lot of kind of low-hanging fruit that, ooh, these animal communication people have come up with all these patterns. Let's see how many of them might conceivably apply to plants. And one of the things that animal people have found is that when predation risk is high, alarm calls of birds, so these three species of birds co-occur in the same forests. When predation risk is high for these populations, their alarm calls have converged to a single alarm call so that individuals of all three of these species can understand the alarm calls of each other and seek protection when a predator is nearby. When the risk of predation is low, alarm calls do not converge, but each of the species, and in fact, often not only within species, but between individuals of the same species, these calls might diverge. And so we wondered if something similar might be going on for our sagebrush plants. So we have, for many of these populations, we have pretty good estimates over several years of what risk of herbivory is, how much herbivory these populations experience. And we find sort of an intriguing pattern that's similar to the bird pattern, so that when risk of herbivory is high, so along the x-axis there's the proportion of damaged leaves. So on the right-hand side of that graph are situations or populations that experience a lot of herbivory. And when that's the case, there's less chemotypic diversity, just one or two chemotypes. When risk of herbivory is much lower, those are the populations where we see much more diversity in chemotypes. And so what we have is the relationship that's shown on the top line. We have a correlation between low chemotypic diversity and high levels of herbivory. And so our thinking is that, ooh, if this is similar to the bird example, maybe high levels of herbivory select for, drive low chemotypic diversity. All individuals can then communicate with one another. And if this is the case, being rare, being the odd chemotype out might be really disadvantageous when you're in a situation where risk of herbivory is really high. And this is a hypothesis that this is kind of what I want to focus on in this coming field season, finding a bunch of populations, documenting risk of herbivory, and seeing if this relationship holds for rare individuals. It's also quite possible, since this is just a correlation, that we've got causation backwards, that in fact, those populations that have low chemotypic diversity are really good for herbivores. So that this scenario about herbivory shaping the diversity of chemotypes or the language of communication is simply backwards, is simply wrong. But anyway, this is where I'm going with this this coming season. So let me conclude. This is my punchline. I hope I've convinced you that communication happens for plants, that plants are capable of a pretty sophisticated behavior, both perceiving environmental cues and responding to them in a way that results in communication. But communication does not happen randomly. It's most effective among genetically identical tissues, among genetically related kin and among similar chemotypes. And with that, I will stop. And I've got, I think, a few minutes for questions. Is that right? Yes.
Rick, it seems that is used on gastric plant. You know if the gastric plant on emitting other times, or is it inducing some sort of internal vascular system?
Yeah, so the question was, we know that, or I hope I've convinced you that the assay plant is getting less vivid when it gets diffused. And the question is it then producing chemicals that are making it more resistant, or is it re-emitting volatiles, if I've got that right? And James Bland has found that the plants that plants will re-emit some of these volatiles so that they can stick to the surfaces of the plants and then be in